it's time to start webinar. I'm Jensen Lim, the co-founder and CTO of HITS. I'm really happy to be here and thank you for joining our webinar today. Today, I will talk about beyond AlphaFold, how far has AI-driven drug discovery advanced? This is content of my talk. First, I will briefly introduce video interviews of Jensen Huang and Demis Hassabis to explore why these two influential figures are focusing on biotech. Next, I will talk about AlphaFold 3 and how AI can solve protein structure prediction, which is one of the most complex biological problems. Then we will see possibilities of combining structure prediction AI like AlphaFold with generative AI. Finally, I will wrap up with a Q&A session. So let's go to our first topic, why Jensen Huang and Demis Hassabis are so excited about biotechnology. Jensen Huang and Hassabis had an interview and talked about AI in drug discovery. In this interview, Jensen Huang made a surprising statement that there's no need to become a programmer. Actually, he was a programmer. If I were to start my career again, I'd study human biology. The comments highlight the importance of applying AI in biotechnology. Demis Hassabis, the founder of DeepMind and Isomorphic Labs, have a similar perspective. He predicted that we will see more AI-designed drugs within 3 years. Their focuses on biotechnology, particularly drug discovery, stems from the belief that as AI advances, it can solve increasingly complex problems. Among these problems, human biology and drug development would be one of the most impactful areas. So let's go into the AlphaFold 3, one of the most famous AI algorithms in this field. I believe the level of difficulty is appropriate for beginners to understand. Nevertheless, there might be some difficult parts due to the technical content. In such cases, please don't feel too burdened and just listen with an open mind. The challenge of predicting 3D structure from its amino acid sequence has been a long-standing problem in science for the past 50 years, and many researchers have attempted to solve it using various methods. The difficulty mainly originates from the astronomical number of possible conformations that a protein can have. There is an example description of proteins, really small proteins. For instance, a typical protein has 300 to 600 amino acids. And for that size of protein, the number of possible 3D conformations is around 10 to the 300. It's a really huge number. And these huge numbers comes from the rotator bonds in each amino acid, which can create countless combinations. If you see this image, there is many rotation bonds, and each rotation bond can have a really large number of possible values. So if we combine these angles, we can create countless combinations. So our goal is to find the conformer at the global minimum. If this is an energy landscape, the global minimum is here. And finding this global minimum in a multi-dimensional space is incredibly challenging in terms of computational cost. Also, not only computational cost, but also accurate prediction of each 3D conformer is very challenging either. These are the main two reasons why many scientists have suffered from predicting 3D structure of protein. And this is why AlphaFold is so remarkable. AlphaFold solved the problem with deep learning, and this is the result from AlphaFold 2, published in 2021. In this left figure, the green is the experimental structure and the blue is the prediction from AlphaFold. As you can see, they are really similar and it is almost indistinguishable. And if you see the right graph, the higher the score is, the more accurate the predicted structure is. When the score reaches 90, the accuracy of the prediction is comparable to resolution of experimental results. The score of AlphaFold 2 is above 80 and is close to 90. So it means that the accuracy of AlphaFold 2 is close to that of experiment. I think the most crucial aspect of AlphaFold is pattern recognition. To understand the principle of AlphaFold more intuitively, I have prepared a simple example problem. Imagine you are a deep learning model and I'm showing you a training data. In the left figure, you can see the various shapes of trees, and these are essentially the typical shapes of trees we can observe in nature in our daily lives. Using these figures, I'm giving you a problem. The tree structures are as shown in the right figure, and I ask you to change them to what you think the most stable form. You can easily find the answer without much thought. You might think of an answer like this. To solve this problem, you probably didn't think about Earth's gravity or various physical processes in trees. You can intuitively know that this is a much more correct answer than the tilted one. This is a kind of pattern recognition done by human intelligence. Similarly, when solving the protein structures prediction problem, we can see many well-folded crystal structure. Then if we give a randomized 3D structure, the deep learning model can turn the randomized structure into the stable structure in a similar way as we find the correct direction of the tree in the previous slide. And what makes this kind of pattern recognition possible is deep learning techniques. Before deep learning, people originally tried to solve this problem by accurate calculation of energy and finding the 3D structure with the lowest predicted energy. If we accurately do pattern recognition, we don't need to solve it with energy estimation. This can be considered the most important change in perspective. And let's go a little bit deeper. I think there are 3 important key principles in AlphaFold. I will try to explain one by one. The first one is prior knowledge, utilization of prior knowledge. One of the most important pieces of prior knowledge is MSA information. MSA stands for multiple sequence alignment. For instance, if a sequence is given, there will be many similar sequences from the different species. And by aligning these sequences, we can analyze their similarities and differences. This image demonstrates the MSA results. And we can see that there are conserved regions and changing residues, which are colored by yellow. And for the purple and mint part, you can see when this specific residue changes, the other side of the pair also changes accordingly. From this observation, we can infer that they are likely to be in similar positions in 3D spaces. We call this as coevolution. So when we have a backbone arranged in 1D, we can infer that these 2 positions will form a closer pair from the MSA information. And through this process, complex energy landscape become much more simpler, and then we can more easily estimate the 3D structures. The second key thing is featureization. The featureization is that deep learning model automatically find key features from the given data to achieve the goal. In this case, the goal is to predict 3D structure. In AlphaFold, there are 2 main ways of featureization. The first one is from the MSA representation, and then second one is from pair representation. In the model, each residue is represented by a token. For constructing pair representation, we need to consider interactions between three tokens. So we can make arbitrary IJK pairs of the token residues. During the training, the model can learn a relationship in spatial arrangement of or variety types of relationships between the three pairs. Through a series of deep neural networks, the model extracts features and ultimately combining them to make a final features. In general, the extracted feature is not human interpretable. This is the reason why we cannot understand AlphaFold prediction. And using this patterned information, the model finally predicts the structure. And the final key contribution of AlphaFold is 3D coordinate prediction. We already know the backbone and its length from the order of amino acids. Then what we need to find is angles between residues like this. So the AlphaFold solved this problem by rotation and translation, utilizing the features obtained from the previous process. In addition, the deep learning model tried to generate 3D coordinates satisfying the triangle equation. Triangle equation is a rule that the distance between 3 points must satisfy. So this constraint dramatically narrowed down the possible conformations, so we can easily find the correct conformations. It is constraint that the produced coordinate should satisfy. And this kind of task is relatively difficult for humans to do. But deep learning can solve this problem through tremendous computing power. So this is a brief explanation on AlphaFold 2. So let's move on to the AlphaFold 3 and let's see what's the difference between 2 and 3. The biggest difference between 2 and 3 is that the 3 provides various multimodalities. For AlphaFold 2, the model can predict only 3D structure of single sequences. But 3 covers not only protein, but also DNA, RNA, small molecule, and covalent modifications. In terms of model architecture, part 1 and part 2 is quite similar, and a little improvement and achievement is applied. But for part 3, coordination prediction changed a lot. The part 3 utilized a new deep learning model called diffusion. So let me briefly explain what the diffusion model is. So just as ink diffuses in this form, if we have a final result image, and we can create a completely diffused state by adding noise to it. And then, like playing a video backwards, we trained AI models in a way that it restores the original image from the noise state. In other words, the diffusion model tried to learn how to denoise the added noise from the original image. So the power of diffusion model is it allows to generate very fine images, which wasn't possible before. This is the YouTube video. This is actually a generative AI based on the diffusion model called Sora, created by OpenAI. Let me start. Okay, this is simple images, and this is much more accurate than the previous images. So AlphaFold has adopted this kind of methods, and by doing this, they reported that they were able to find atomic positions very precisely without the need for post-processing like molecular dynamics. So in the end, AlphaFold solved the protein 3D structure prediction without using the energy landscape information, which I mentioned earlier. And it is really meaningful that this has overcome the fundamental limitations of existing physics-based methods. This is a result on performance of AlphaFold 3. Let me briefly explain. Please see this graph. For example, the accuracy of post-prediction between protein and ligand is about 76%. If we see the existing other deep learning model named RositaFold, it's about 40% accurate. So it shows about twice the better performance than the RositaFold, and it shows a far superior performance than the docking programs like Autodoc. The important thing is that it can find the structure when only the sequence and ligand structures are given. We don't need the predefined 3D structure of protein, which is necessary in conventional docking method. Moreover, it can be applied to nucleic acids and covalent bonds, and then it can predict various structures such as antibodies, protein-protein interactions, and so on. And these amazing changes have been made possible. And as a result, we can find protein structures with AlphaFold alone, even if we have only sequences. While scientists previously could find hundreds of thousands protein structures through X-rays. And it took several decades to achieve these numbers, but AlphaFold generated about 2 million structures less than a year. It's quite amazing. And if you go to this database here, and you can easily search for the structure of your problem. So we can say that it has predicted the structure of almost all proteins on Earth. In recognition of these achievements, Demis, Hassabis, and Jun Jumper, the key authors of AlphaFold 2, received the Breakthrough Prize last year. For your reference, this prize has a larger prize money than the Nobel Prize. It gives the world's largest prize money. It's about $3 million for each winner. And this gave confidence that really complex biological problems like predicting 3D structure can be solved using deep learning. Additionally, through AlphaFold, numerous AI researchers with backgrounds in mathematics, computation science, or physics have moved into the biological domain. For instance, Demis Hassabis actually majored in computer science, and Jun Jumper majored in physics and mathematics. And it's interesting that these individuals received the Breakthrough Prize in biology, even though they are not majoring in biology. As an example of this trend, I analyzed the statistics from the last year's NeurIPS conference. And NeurIPS is the largest artificial intelligence conference. There are 3 major AI conferences: NeurIPS, ICLR, ICLR, and ICML. And NeurIPS is the largest one among 3. And upon analysis, we could see how much of hot topic drug development has become in the AI community. A total 300 and 500 papers were presented. And among them, there were 124 papers from drug and 156 papers from protein, and many others are related to biology, molecules, and antibodies. And many papers are being published of this drug development related keywords. Please remember that this is an AI conference, not a biology conference. And you can see the similar trends in not only at NUIX, but also at ICLR, ICML, and other AI conferences. More and more bio-related papers are being published. For those interested in more details, you can check our Hyperlab blog. And there's another implication that when paper of AlphaFold is published, the authors didn't release the code. It's quite impressive. I mean, surprising news. Up to AlphaFold 2, they had been releasing their code, so anyone could use AlphaFold 2's code. However, for AlphaFold 3, they didn't release the code, and this caused quite controversy between scientists. And one of the main criticism is that DeepMind used the public PDB dataset created over decades with public funds to build their model, so they need to open their model. We guess that the reason of not releasing code is potentially for commercial purposes. As a result, many people declared that since the paper is out, they could create the code themselves, and currently many people are working together to create the code. In fact, Nature News reported with the headline, "Who will make AlphaFold 3 open source?" Scientists races to crack the AI model. So many people are working on it. And another significant implication is that solving this problem is really expensive. It requires an enormous workforce. The average annual salary for a single PhD of AlphaFold 3 team would be $500,000. AlphaFold 2 had 48 authors. So from this number, we can estimate the expense of money. And this is no longer just a business of ideas. Business is fundamental, and according to the newses, the effort to create the code is only 5%. The rest is money. Conservatively estimating the money used to develop AlphaFold 3, the salary expense alone was about $74 million. And if we include learning cost, I mean computational cost, we can expect it to be much more. As you know, the computational cost, especially GPU, is increasing dramatically. So I covered about the half of my presentation. And if you have any questions for this part, please leave them at the chatting. And I would be happy to answer them at the Q&A session. So then let's move on to the next topic. What becomes possible when predictive AI and generative AI are combined? The first application I'd like to introduce is a model named DiffDoc. Here they also utilize the diffusion model I introduced in my first part. The two authors of this work majored in computational science. Is also interesting. And in this work, the 3D protein structure and 2D ligand structures are given to the model. And the task of the model is finding the correct binding structures between ligands and proteins. In the video, if you place a ligand anywhere, then the model predicts where it binds in a way of blind docking. In other words, if we don't know the binding site information, we still can predict where the ligands would bind. This is also quite impressive because in many previous conventional methods, we needed to specify the binding site for efficient search of binding conformers. And these kinds of model can be further improved for induced fit docking. When a ligand binds to a protein, the protein structure actually changes. proteins are not rigid and fixed. And in the work shown in the slide, in this work, they use deep learning to simulate this induced fate effect. The induced fate effect, the structure changes to a hollow structure. They also utilize the diffusion model. And before this kind of model, molecular dynamics was the only way of capturing this effect. However, the fundamental limitation of molecular dynamics and other physics-based simulation methods is that is the tremendous amount of computational cost to simulate proteins to see meaningful structural changing. But the new deep learning model named dynamic spine can answer it within minutes with much small computational cost. And the approach is quite similar to AlphaFold. The model also doesn't use energy prediction. It's free of energy estimation and searching conformers. So instead of following physical energy landscape, the model can find the desired answer by following a pattern recognition, which is used in AlphaFold 3. And the next example would be more interesting. It is the combinations of high accuracy structure prediction and generative AI. In general, you need activity data to design biologically active drug candidates. But the problem is that activity data is hard to obtain. It takes a long time and money to enough number of data to train deep learning models. So one challenge is whether we can design potentially bioactive molecules through AI without expensive activity data. So the model proposed by Uyen Kim's group in KAIST, who is also CEO of HITS, demonstrate that it is possible to directly generate a new molecule in 3D space without activity data. Even more, binding poses in this 3D structure can be generated either at the same time with the 2D structure of 2D molecular structure. And let me show you the video. And there is a protein, and deep learning model make a new molecule as like 3D printing them in the binding pocket. And if you see the right graph, you can see that when starting with the random scaffold, we can design ligands that can bind quite effectively to new proteins that weren't trained on without any activity data. This red dot is binding affinity of the scaffold, and this is the distribution of generated molecules. And this distribution is located below the red dot, which means that the generated molecules have higher binding affinity than the starting molecule. More recently, in his lab, he published quite influential paper using diffusion models to design drugs that fit the pocket. The main achievement of this model is that the model considers the pharmacophore information of the pocket for molecular design. For the tight binding between ligands and proteins, it is essential that the ligands make specific interactions with protein residues. So consideration of pharmacophore information can be beneficial for designing more effective binding ligands. The diffusion-based generative AI he developed his diffusion-based generative AI not only predicts structure but analyzes the pharmacophore around the protein's binding site and creates something that fits that pharmacophore. So let's see the video. And the deep learning model is in process of making a new molecule from the randomized structure. For instance, if there is a source for pi stacking or sort bridge, a matching substructure is created nearby the source of interactions. One important application of this model is designing selective inhibitors, which is also a really challenging task in many drug discovery projects. What he tested in this paper was EGFR and mutated EGFR. The wild-type EGFR and mutant EGFR have different residues in the binding site, so naturally they have different pharmacophore in certain parts. He showed that whether drugs designed drug using these different pharmacophores actually exhibit selective binding. The parts we've shown in blue are what we designed, and you can see they are bind strongly to the mutant but relatively weakly to the wild type as desired. But on the other hand, red graph is from general diffusion model, and there's essentially no differences. Next, I will show you another example. This example is from protein design. And this paper was introduced in Nature last year and was published by one of the most famous researchers in this field, David Baker. The Baker's research team utilized diffusion models here. And what they did was to show the design of new drugs of various modalities is possible. So here is demo videos. As shown as a first example, given a target protein. In this case, target protein is a small peptide. If I ask to create a protein that can bind well to the given peptide, this diffusion model designs a protein with a new sequence that binds well here. And in second example, the insulin receptor is given and a specific binding surface is specified. And the purpose is designs a new protein that adheres well to the surface. So through this diffusion-based generative AI models of various modalities, it's showing the possibility of designing new proteins or antibodies. And many researchers are working on this project to further develop this kind of models. And for the next, so it's quite curious that how well do this AI actually work in experiments, and there are relatively a few number of experimental reports, and I prepared one of our case. We actually used our AI models in collaborative project with Korean biotech venture. First, we started with screening. We did virtual screening of 800 and of 1,000 compounds using our own AI. And we derived 40 compounds and found 3 hits through experiments. And since these can be bought commercially, we obviously can't patent them. The next goal is to find hits with novelty that can be patented. Through hit lead process, we newly designed and synthesized 14 compounds and found 3 hits among them. And we confirmed the actual anti-cancer effect through a xenograft model. And finally, we found 2 lead compounds that could be patented. As you can see, we were able to find lead compounds through a very small number of experiments. The number is quite impressive. And there are some more experiments reported from David Baker's lab, and they reported cases where they experimentally tested newly designed sequences by their deep learning model named RF diffusion. Next, instead of difficult technology, I'd like to introduce 2 interesting but opposing views on AI in drug discovery. The first is from Drug Discovery Today. This is a review paper, and the review paper was written from the Boston Consulting Group, and it is one of the world's leading consulting firms currently. And to summarize, there are two main points. First, in the past 5 years, increasing numbers of drugs and vaccines have been discovered, and they are entered clinical trials. And the graph below shows the number of cases entering clinical trials per year. And there are several categories: AI designed drug, AI discovered targets, small molecules, antibodies, vaccines, and repurposed drug. As of 2023, there are about 67 in total. Looking more closely, a drug discovered by AI showed a higher success rate in phase 1 clinical trials compared to the historical success rate of drugs discovered through non-AI methods in traditional way. And out of 66 cases, and 42 are currently ongoing, and of the 24 that have been completed clinical trials, 21 were successful and 3 were failed. Of course, there are people with critical perspectives here as well. If you look at this science blog, there's a quite famous medicinal chemist named Demis. He has worked as a medicinal chemist in many global big pharma companies for the last decades. He wrote an interesting essay with a critical perspective on the review paper we discussed in the previous slide. The title is AI Drugs So Far. In this essay, he said that among the 24 targets discovered by AI, which mentioned in that review paper, claiming that AI's many successes, among 24 targets, all except 8863 were already known. This one might be, 8863 might be considered somewhat new, but the last were all already known, and even more, some were in clinical trials or already on the market. That's what he's saying. Then regarding the statistics showing that AI-based drug discovery performs better in clinical trials than traditional method, he argues that the sample size is too small to make a significant conclusion. He said that it is not statistically meaningful. Then another point is that among the 114 AI startups analyzed in the previous paper, 80% were not actually AI companies, but just regular biotechs. So there's an issue with the credibility of this survey. In other words, we need to investigate how much AI was actually used in each project and how much wasn't. That's what he's saying. So I'd like to summarize today's talk. To summarize, AI is clearly enabling big things in drug development that weren't possible before without AI. In certain areas like AlphaGo 3 or drug design, accuracy has been improved significantly compared to the past. However, the prediction accuracy is still lower than the experiments. As you know, experiments give us definitive answers, while AI can replace some experiments like predicting 3D protein structures. However, it's still not accurate enough. The AI cannot fully replace experiment in most areas. Nevertheless, given the current pace of development, I believe that some problems could be solved within a few years. Looking at some cases that have entered clinical trials, we can see that we need about 200 to 400 molecules for designing, synthesizing, and experiment. To enter clinical trials. Traditional drug development typically synthesizes about 800 to 1,000 compounds. So roughly saying AI is about 2 to 5 times more efficient. I think it's a quite impressive result. However, there are no cases yet where an AI-developed drug has passed all the clinical trials and been approved. And that's the current reality. We need to clearly see the current reality. And as I mentioned earlier, while certain areas are developing rapidly, some are not. One of the examples is predictive performance for toxicity, side effects, immunogenicity, etc. is not well known. It's still a question mark. To address this, I think AI needs to evolve to predict drug properties by combining not only drug structures but also multi-omics data and other various modalities. Actually, some companies are working on this technology, but it's not showing as impressive performance as AlphaFold 3 in protein structure prediction. And another major challenge is the improving performance in clinical trial. And to do this, we obviously need to use clinical data and how to integrate clinical data from hospital is another important change, and we still don't have clue yet. And lastly, beyond small molecular protein, we need to expand to more interesting areas like mRNA or cell therapy. And these are gaining attention as new types of modalities. This is another challenge we face. And the next section, which is the final session of my talk, is Q&A. And several people ask questions, and I will try to answer to these questions. The first question is that, will AI drug discovery play a role in pharmacogenetics? Actually, I don't have extensive expertise in this field. To the best of my knowledge, pharmacogenetics is not the most active research area where AI is actively applied, but quite many researchers are working on analyzing huge amount of multi-omics data with AI. It may not gain significant attention in the short term, but in the long run, like other scientific fields, and it could be greatly influenced by AI. And I believe this is the correct direction, as I explained in last part of my talk. And the second question is, does post-AlphaFold help us find the complicated biostructures like proteins? My answer is definitely yes. AlphaFold 3 has already demonstrated its ability to accurately predict not only protein structures but also RNA, DNA, and small molecule interactions. In the future research, will rapidly advance beyond the structural prediction, but to the design of biomolecules with desired 3D structures. And the third question is, do you think that the success of AlphaFold could have implications in protein-protein docking for applications in the product design? I believe that accurately predicting protein structures will greatly help in protein-protein docking as well. Besides the predicting structures, predicting binding energy remains a challenge. It is not accurate as structure prediction now. And in addition to structure prediction, the field of protein structure design also advancing really rapidly. I tried to answer those three questions as well as possible. I hope my answer was helpful. And this is the end of my talk, my prepared talk. And I really appreciate your attention to my talk. And if you have any more questions, please ask via chatting, and I'm happy to answer them. Thank you very much. And I will wait a minute for the questions. So I will wait a few minutes to have questions. I will wait about a minute. It's 11:14 in Korea, so I will wait about to 11:46. Okay, I think there are not questions anymore. So after the my talk, if you have questions, feel free to contact me. And so then we can stop here, and really appreciate your attention again. Thank you.